So we're going to continue working our way along this timeline. And what we've had so far essentially is look at the origin of some of the biological molecules from inorganic molecules, how they may have then given rise to RNA molecules that would be self-replicating and then packaged up inside protobionts, so pre-cell type structures. Those would give rise to the first cells around 3.8 billion years ago, which would then be our prokaryotic type cells. And so our prokaryotic cells really then are branched into two domains, which we'll get into as we talk about classification and taxonomy and so on. But we have our archaea and our true bacteria. What we then looked at was the selective forces, essentially the force being oxygen. So photosynthesis is really the really important process here. It's the driving process that then creates the oxygen in the Earth's atmosphere. That becomes a selective force on cells that can't stand being around oxygen, or those cells that have an advantage because they can actually use oxygen. And those cells then proliferate. Those cells then also form relationships with other cells. So we have endosymbiosis, or symbiotic relationships between prokaryotic cells, and that gives rise to a new type of cell, a more complex cell that is our eukaryotic cell. And so eukarya is another domain. So we actually have three domains, right? Archaea, eubacteria, and then eukarya. So this group here is where we find animals and plants and fungi and the protists and so on. Okay. So the origin of them would start here around 2.1 billion years ago with the first eukaryotic cells. What we're going to look at now are how some of those cells, some of those single cells, would have then come together to form multicellular structures and give rise to multicellular fungi and plants and animals. And some of the protists or algae that are also multicellular, and give you a few examples from today, from actual living things, and how they behave and how it kind of reflects a potential history of their ancestry becoming multicellular. So to give you some more dates here as well and go along the timeline, so what we have is for multicellularity, multicellular organisms dated through common DNA. So common gene sequences within our DNA, things that we share with other multicellular organisms, can be dated back to about 1.5 billion years ago. So potentially somewhere around 1.5 billion years ago is when some of the things we're going to talk about started to happen, where some of these eukaryotic cells started to come together and form more complex structures, or aggregates of cells that then gave rise to differentiation between having cells have different jobs, and then that gave rise to multicellular organisms. And again, we'll look at a couple examples here, example with sponges, an example with slime molds, to kind of see how that could have happened. We have in the actual fossil record, so fossils around 1.2 billion years old of some multicellular algae. So we know that it's definitely around that time that some of those single-celled organisms started coming together and forming multicellular structures. And these are all very simple sorts of things that we find evidence of in the fossil record. Up until about 535 million years ago. So between a time period of 535 to 525 million years ago, an event called the Cambrian Explosion occurred. What does that mean? Well, first off, Cambrian, it's one of the geological time periods that we discuss. So when we look at it, the timeline like this, you can expand it out and actually assign different names to the different times. and this is the time period called Cambrian. And it's called the Cambrian Explosion because before this time period, we see a certain evidence of organisms that existed in the past. And after this point, the diversity of organisms rapidly expands into a variety of different groups, reflecting some of the groups that we have today as modern groups, all coming about right around this particular time. So we said back when the eukaryotic cells formed, there was a selective pressure which we could find evidence from as oxygen in the atmosphere. And so, same thing, at this time, something would be happening with the organisms, their diversity, their the number of organisms, populations of organisms, something that would allow new types of organisms to form and then those types to thrive. Right? And that would have led to this Cambrian explosion. So really before the Cambrian explosion, some organisms we see, say in terms of animals, we see more simple animals like sponges, which are the porifera, cnidarians, which are organisms like sea anemones and corals, and mollusks. Mollusks are a diverse group, having some more simplistic members of the group and some very, very complex members like the cephalopods in the group, but we're talking about here more the simple chitons and things like that, which are not as complicated as an octopus or a squid. And then after the Cambrian explosion, though, is when we see those types of organisms coming about. So some of them being within the same group, so like the mollusca, this particular phyla, which we'll cover again later in the course as we look at diversity, goes from having members of the group that are just having these simple forms, and then those forms then changing and being modified into a variety of other adaptations, and some of them to get very complex. In addition to that, whole new groups of organisms come about, a variety of a lot of groups of organisms. And many of these are now more complex in structure, and many of them have different behaviors in that they are predators. These organisms here are more, like I said, simplistic in form. We'll talk a little bit about form, but they're also they're more filter feeders. They filter feed on particles in the water, pulling it out, or they're scavengers, or herbivores eating algae. But there are no predators really in this group. And then after the Cambrian explosion, there's this dynamic change. So probably competition as well is probably one of the things that drove that change for space, for resources, and then those organisms that can adapt to that likely started to do. And these are all things we'll go over in different parts of the course. Again, we're right now just really more on timeline, and I'm just adding in some interesting side notes or some interesting points to that and some of the things that I want you to know specifically about the times. So, but how would this have happened? So with some of the other organisms and the other events we talked about, I gave you some evidence, as far as like, well, but can we explain this process? So the process of multicellularity, what I'm going to do is briefly go through a life cycle for an organism called a slime mold, and not the whole thing, but the basics of it, and then some of the structure of a sponge, a porifera, and to kind of give us a sense of how multicellularity could have come about. So first we'll look at the slime mold. And there's a particular genus that you can look up. I'm going to just get rid of that one. Called Physarum. Okay, so Physarum, if you look up a video on it, it's very kind of freaky and really interesting, cool, because what you'll see are single-celled amoeba. And so these single-celled amoeba would be like our, eukaryotic cells, single cells. These amoeba live on their own, doing their own thing, finding nutrients and dividing. They can then get a signal. And that signal causes an aggregation. Usually it's nutrient related. And what will happen is that aggregation will cause, well, the signal will cause the aggregation, and this aggregation will essentially be hundreds of these amoeba all joining together to form this blob called a plasmodium. And it's mobile. It can crawl around. So it's this multicellular structure. It's a slug-like, so people often call it slug-like structure. And again, to really get a great sense of this, you should look up, like I said, watch a short little video on the plasmodium moving around. So this is a multicellular structure that forms in a response to a chemical signal in search for food. And then what will happen is this plasmodium will move around on its own, right, looks like a worm-ish structure, kind of moving around until it finds a particular space to begin the process of forming spores. And so when it does find a particular space to do that, it'll start to form a fruiting body. And the fruiting body will give rise to spores. The spores can then be distributed, and spread out. So again, this is sort of a response to starvation and now finding new food sources. So food sources have run out, so the organism is now needing to distribute these spores all over the place. Hopefully, some of them land in a place where there's food. These spores will then develop back into little amoeboid cells. And those amoeboid cells will just be single cells that just crawl around on their own, and they can do this. There's other parts of the life cycle which we're not going into, but the point of this was to give an example of how single cells can do their own thing and then be given specific signals and then form into a multicellular structure that all behaves together. Some people have actually done studies showing how this multicellular plasmodium can navigate through a maze based on sensing chemical signals in the environment. So it's strange that it's just single cells, but then they actually work together, communicate with one another, can sense the environment, and respond behaviorally to changes in the environment, still just being a bunch of single cells that are just grouped together. So that is something that's an interesting example, and this is an organism that we have today that is alive that we could see and people study, and it displays this behavior. So it's a type of protists that is, to say is it single-celled or multicellular is kind of difficult sometimes to peg because of the different stages of their life. The other thing we'll talk about briefly are sponges. So sponges now are animals. And so they're in phylum Porifera. Now again, we'll go over more details of phylum Porifera later. But the sponges have a cellular level of organization, meaning that they don't have true tissues. Right? So sponges have these special cells called choanocytes, which are these collar cells. And they are a filtering cell. So they have a flagella here, and again, we'll look at those. And they create current. So water current moves. It moves through the collar cell. The collar cell is actually like a mesh, sort of like web, and they can capture, little particles from the water, and then phagocytize or ingest them. So these are sort of a food capture mechanism. Behind them are amoebocyte cells that then can take nutrients from these choanocytes and then move around and distribute them to other parts of the sponge. So when a sponge, let's give a quick layout here. When a sponge forms, its body is essentially layers of cells, but they're just separate types of cells that work together for some common goal. And so what we have are sort of chambers within it. Those chambers are lined with these choanocyte cells, these collar cells. I'm oversimplifying it here. There actually be hundreds and hundreds of these cells lining these compartments. But the idea is the sponge forms this filter-feeding, maybe a vase-like structure in some cases, where food particles can come in. Some group of cells collect the food particles. They pass them on to other cells that then move through the interior layers of the sponge and then distribute the nutrients to other cell types. There's a whole variety of other types of cells we're not going over now, but we will later, that show sort of a collection of cells of different types. Now, the other thing that's very interesting about sponges is that you can take a multicellular sponge like this and in a lab put it in calcium-free seawater. So seawater has calcium chloride and sodium chloride and magnesium chloride and a variety of different salts in it, not just sodium chloride, but a variety of other types. If you were to make up salt water and use all these other types of salts except no calcium, what we'd find is that these cells will then fall apart. And you'll get little panacocytes and choanocytes and amoebocytes and all these different cell types just sort of separate on their own because they use certain binding proteins to hold themselves together, and they're not really true tissues. So take this animal, put it into seawater without calcium, it just falls apart into a slurry. It looks like it's dissolved or dead. But if you look at the cells, they're actually still alive. And then one of the very cool things that can be done is that you can collect up these cells, put them then into seawater that has calcium, and the cells will start to re-aggregate and come back together again and reform the structure of the sponge. Okay, so they will then re-aggregate and organize. So again, living sponges today, we can actually see this in a lab where single cells can behave separately as individual entities, but then they can collectively work together to form a multicellular mass that then has different jobs, and that would be selected for because then one cell doesn't have to do everything. Like all the cells don't have to collect food. Certain cells' job is just to collect the food. Other cells can distribute the food. Other cells can provide a defense mechanism. Other cells can carry out other things. And then as an organism becomes more complex, it's going to need probably more cell types to carry out more sorts of jobs. All right, so our origins of multicellularity, we have the timeline sort of roughly like when this happened. And we have some ideas of how it happens and some evidence today from examples of organisms that have both a single cell and multicellular form and that collectively come together. And so then we're from that point on, we're kind of getting up into organisms. And so the next thing is we'll start to really just talk about diversity. And so we'll start going through all the different groups of organisms and their characteristics.